This afternoon we have our first session is on bioinformatics tools and databases, and I have the pleasure of introducing myself first. Today we're going to hear from myself first, and then from the CRG we've got two talks from Jose and Bjorn. Okay, great. So today I'm Peter Harrison. I'm the Genome Analysis Team Leader from the European Bioinformatics Institute, and I'm going to give you an overview of the EuroFAANG Data Infrastructure and then how we use that to standardize and present both BovReg data, but also global FAANG data. So the European Bioinformatics Institute, our mission is to help scientists around the world realize the potential of big data. We do this through providing freely available data and services, through our cutting-edge research and technology, and then also through a huge amount of advanced bioinformatics training. So really we're providing the services and expertise to support the coordination and archiving, the annotation and presentation of BovReg and FAANG data at scale. So since 2015, sort of very close to the start of the FAANG project as a whole, and which is now celebrating its 10-year anniversary, we formed the Data Coordination Centre at EMBL EBI. It was first funded by the UK government and has since been funded very generously by the Horizon projects within Europe. And it really supports FAANG and its aims of trying to create data openness, reusability of data, to standardize it, to create this harmonized, rich genome to phenome resource. So we're trying to ensure that all of the data is richly described, it's available, and it's searchable, that we can present it consistently to all of the community, and that we can facilitate the standardization both of data formats, of the protocols that you provide with your data sets, and then also more recently, and a lot you're going to hear from Bjorn and Jose, on the standardization of analysis methods as well. So really this is all about making sure that all of the FAANG data is open, that it's reusable, and that we can really ensure that we can have these large-scale cross-project meta-analyses. I'm going to briefly introduce EuroFAANG. You're going to hear more from Klaus tomorrow on the EuroFAANG research infrastructure. But this was where we actually brought together the Horizon-funded EU projects in order to standardize their research processes, the analysis and methods that they were performing. So this is a hugely collaborative effort and a very important effort that's happened within Europe, and really has increased the effectiveness of all of the Horizon projects that have been part of this collaboration to help them collectively improve the animal production, welfare, and the research they're doing on supporting industry. So really what this is able to do is actually collectively leverage expertise so that people across these different projects, which include BovReg, have been using their expertise collectively to work on analysis pipelines, to standardize data and work on the same metadata standards, to collaboratively work on improving ontologies, to really improve how we collectively in Europe do genotype to phenotype research. And the data infrastructure that we've developed in these projects is now forming a key component of the research infrastructure that you're going to hear more about from Klaus tomorrow. So, and this research infrastructure is now a key part of this. Is it allows us to continue our efforts to continue to standardize genotype to phenotype research within Europe and then across the world. And we're going to now be expanding this through this infrastructure to new breeding, phenotyping, biobanking, and genome editing technologies. But whilst this has this European focus, which is so important to us, we are still going to maintain this very strong connection to global FAANG data, supporting global FAANG projects around the world. So our role within the BovReg consortium is in two areas. One is around scientific reproducibility, and this is we've worked very closely with the CRG on this, is about the development of standardized pipelines, and our role in this is then the presentation of these pipelines to the community to make them accessible and ensure and try and promote that across European projects people are reusing these pipelines that people have put so much effort into within these projects. But then also on the focus on the open distribution of data. So once BovReg is putting its publications out, we're going to ensure that the community has rapid access to the data and to make sure that it's findable, accessible, interoperable, and reusable. So really this is what makes FAANG datasets so special, is the fact that along with the data file that you can download from the public archive, you get this incredibly rich, consistent, and validated metadata descriptions. So you can very easily understand what experiment was performed, what sample that you're looking at a data file from, rather than just being provided with a rather obscure name and potentially a location, and that's all you get. With this, we have these incredibly rich metadata standards that people put a lot of effort into curating. We then also, within FAANG, have standardized core assays and agree on what histone marks, for example, that people will generally produce data on. We also have these very rich mandatory sampling, experiment, and analysis protocols that you have to provide with every dataset, which gives almost an incredibly in-depth description of how any sampling process, how any experiment's been performed, and how any analysis has been performed. And then now we're also requiring you to provide your analysis workflow along with any data submission you make as well. And that was a change that came into effect within FAANG just last year. So really this is this community drive to ensure that this rich genotype to phenotype data is open and it's fair. And then alongside that, we're then trying to develop new data presentation and visualization methods to then try and present this data effectively and easily for people to reuse in their research. So if you take any given data set within FAANG, you'll find that it has this rich metadata. It's got these standardized and mandatory protocols. You have pre-publication of many of the raw data sets within FAANG. You have this automated literature tracking, so we understand the impact and how this data is being reused. You have attached to it workflow pipelines, particularly the NF core analyses that you're going to hear from Bjorn and Jose about, which are so important for our standardization of analysis within Europe. And then also you'll have a standardized ensemble annotation and mapped tracks, as well as the community annotations that have been done by the projects themselves. So really, this sort of metadata solution that we have for FAANG is really trying to aim to ensure that any given FAANG dataset is easily understandable, can be repeated, and can be reused. And that we're using very consistent terminology, so we have a huge focus around ontologies, and we structure the data and that it's rich and specific. And this is really how we're trying to ensure that any data set that's produced by using public money can be reused multiple times for years to come. And a sort of a key part of this is standardizing the ontologies. This is the way in which we control the descriptions of the metadata. And this is why it's important that these ontologies are so accurate. So in ontologies, if you don't know, and we kind of use these to sort of describe any biological entity. So each one of these sort of terms has a unique identifier, which computationally allows us to then use these in a structured way in order to present you to data, in order to allow you to find data more easily. And one of the key aspects is that ontologies have a hierarchy. So if you're describing, for example, a particular location within the brain, we can then allow people to search for more generic terms, such as the brain as a whole, and gather that data and present it back to them when they're running those searches. So one of the tools that we provide at EBI to help people with this is the ontology lookup service, which is a service that presents ontologies that are curated from around the world and allows you to search over those and find terms that are most effective for your research. And the FAANG portal integrates very closely with this service in order to try and validate and ensure that we're using accurate descriptions for FAANG metadata. So one of the things that we've developed in collaboration and through BovReg and through our collaboration with the other Horizon 2020 projects is an ontology improvement tool to try and make it easier for you as the community to improve these ontology descriptions, as they're often very medical or model organism focused, and to allow you to adapt them for use in agriculture. So this allows the users of this service to link it to the project, so we will automatically have loaded all of the ontologies used in any BovReg dataset. It's an authenticated service because part of the importance with ontologies is you have a provenance, you understand which scientist it was that actually proposed this term, that they have the credentials to support that. And then any user of the FAANG service can come here and flag an ontology and say this needs improvement. Then anyone with the knowledge to improve that can then come back and collectively work with others on improving that ontology. And then we will then forward those changes to the underlying ontologies on your behalf to then make sure they're implemented for future use. So if you're particularly interested in metadata standards and making data fair, as part of Global FAANG, we relaunched this year, or last year, new task forces, and one of those task forces is called MetaFair, and this is the task force in which people come and we discuss and we decide on what metadata improvements we make for FAANG globally. So if you have a new large project that you're forming, it would be very good if you send a representative to this task force to ensure that we're supporting what new work you're doing, or if you're working on a new technology that FAANG hasn't used before. So really, we need your expertise to keep improving the metadata standards. So once we've got this very rich data sets that have been submitted to the public archives, we collect them all together and present them back to you as a community in the FAANG data portal. So this is your single access point to all of the wealth of data that's been generated by any FAANG project around the world. It provides you direct access to download the data from the underlying archives. We automatically track reuse of any of the data sets. If anyone puts that data set in a publication, we will track that and provide links to those publications from within the portal. We try and provide as intuitive search and filtering as possible using this rich metadata and these ontology terms. And then we provide visualization and cross-referencing over that as well. So part of this service is the actual submission service itself. So all FAANG data, before it goes to the public archives, has to go through this pre-submission validation. And this is how we ensure that the metadata is rich and all of the descriptions are accurate. And this not only highlights errors, it will also actually try and suggest improvements. So if you've used a very generic ontology term, it might try and suggest that you make a more specific one. But it's a brokered submission service, so we actually then manage the submission of the data to the archives on your behalf, so you then don't have to do that, and it ensures that it's then done accurately. And also through FAANG, you can also submit your protocols, sort of richly describing how you performed your sampling and experiments. You can submit to track hubs, so once you're making your publications, you can provide converted files that are in track hub format, so they can be easily visualized in genome browsers like UCSC and Ensemble and in JBrowse. And then also, now you can also submit your workflows so that we have this, build up this reusable data sets as possible. So part of this is we also provide dedicated spaces for the EuroFAANG project. So BovReg has its own data portal within the FAANG infrastructure, and this is a dedicated space where we present all of the wealth of information that this project has generated. And as more and more of the publications come out from this project, there's going to see more and more data sets appearing here and more tracked publications. But it also contains a wealth of other information generated by the project, the publications, all of the workflows that people have put a lot of effort within this project into generating and curating. And then we will actually, over time, we will automatically track publications. As this data is reused by the community for the coming years, we will continue to track those publications and see the impact that this project has had. And one of the things that we're particularly doing within BovReg is ensuring for every analysis that you've done that we're very clearly linked to what Nextflow workflow or what GitHub workflow generated that data set, so it's completely repeatable. So one of the other things that we have is this TrackHub brokering platform. So this is a way in which you can essentially make your genomics data that you've generated web accessible, and really you can view it within the context of the annotation of the genome, the reference genome, and visualize this data within a range of genome browsers around the world. So we have a TrackHub, a brokered submission tool to try and make this easier for you to provide these. It's built upon Ensemble's TrackHub brokering platform, and it really allows us to register that data on your behalf. And then we also ensure that when you've done that, we'll link it within the FAANG data portal to your other data sets. So when people are looking at your experiment data, they will see you've also produced a FAANG TrackHub as well. And really this is trying to make your data as easily accessible as possible, so that when people can then discover it both directly through FAANG, but they could also just discover it independently by looking at the genome browsers and seeing what track hubs are available for a particular reference genome. So this has a submission platform. We do some validation over that, and then we can also store your data on your behalf. Because one issue often with track hubs is that people put them on their university servers. Those servers then disappear. The track hub becomes unavailable. So we're committing, at least for FAANG, to try and provide a central repository so these will remain available for years to come and for the duration that FAANG is in existence. So one other thing that we've put in place recently is within the FAANG data portal itself, we now have a lightweight genome browser. And this is to allow you as the projects to then present their data. It's based on IGV, and it provides a very simple and powerful way to visualize data within the portal itself. And then you can then go out to your own favorite genome browser or your own downloaded genome browser to do more investigation. So this can be customized for your tracks. So as these publications are coming out, we'll hope to continue to work with the projects in order to represent the data and have them instantly loaded to the view or the point of interest within the genome that's most important to that project. And a key thing here is that you can select your reference of choice. So if you're using a different reference to the one that's currently in the mainstream genome browsers, you can actually present your data against a different reference genome and don't have to then completely remap it every time. So one of the other, with my other hat on, I'm also one of the team leaders for the Ensemble project. And this is a genomics platform and browser for trying to enable and accelerating downstream science. We provide gene annotation, comparative analysis, transcript annotation, a lot of work on variation, and then more recently we've started to produce regulatory builds for animal species as well. So this provides our own standardized and high quality, richly described annotation, and we then also provide a range of tools such as the variant effect predictor, which is very important for industries. It allows them to then look at their points of interest and then actually query these against the genome for what the predicted effect might be. So the sort of annotations as the BovReg data is released, we will continually update the cattle annotation to include the wealth of BovReg data. And as we've seen with the other Horizon projects, we're expecting this to be quite a marked increase and improvement in the quality of the annotation that we will then be able to produce from Ensemble. So the Ensemble annotation already includes some of the cattle data from the US, but we've also got more cattle data from the US being released shortly, and this can then be combined with the BovReg data to produce a very rich and high quality annotation, as well as the ones that are going to be produced by the BovReg project themselves, which we hope will be able to be viewed against this as well when we have GFF3 submissions available in the public archive shortly. So as well, Ensemble in the last year has started to produce the first regulatory builds outside of human and mouse. So we've now produced builds for 5 aquatic species in collaboration with the EuroFAANG project and with the GeneSwitch project, and we've produced a regulatory annotation for chicken and pig. So these have driven new regulatory methods that we've developed within Ensemble, and then also new visualization that we've had coming for this as well. So you now have these regulatory builds viewable in the Ensemble browser, and we're looking forward to hopefully being able to add a cattle one as well once we have the data available from the US and from the BovReg project. So one of the other things Ensemble provides is the variant effect predictor tool, and this allows sort of maps the variants and transcripts and regulatory features and then predicts their molecular consequence. So what that particular consequence might have upon the genome, and allows industry to look at their own lines and then actually look up against the wealth of public data that BovReg has now been producing in order to sort of ask the effect of consequence against the genome. And it can quickly analyze a large set of GWAS hits and then filter and provides visualization over that as well. So in terms of the cattle reference, we essentially use all publicly available data. So as the EuroFAANG projects and the global FAANG projects make their data releases, we will improve our annotation repeatedly over time. So we've just released UCD 1.3, and very shortly releasing in the next few weeks UCD 2.0, which contains the Wagyu Y as well. And one of the other things that we're particularly interested in is continuing to grab and gather more breed annotations as we move towards a pangenomic view of cattle as well. So in terms of what we're looking to do, we're really looking forward to the wealth of data from these projects to continue to become available so that we can improve our own reference annotations within Ensemble. We're looking forward to adding the telomere to telomere references as they become available in the coming years. And then in particular, we're very interested in continuing to gather new breeds and strains of economic and research interest so that we can start to build this pangenomic view as well. And then one of the aspects that we're particularly focusing on is this pangenomic view, and you're going to hear later from Fergal Martin, who's my colleague within Ensemble, about the work that he's been doing in the Human Pangenomic Reference Consortium. And then Ensemble as a whole, we've got an aim through the Earth Biogenome Project to provide a reference annotation for every eukaryotic species on Earth. And that's our sort of long-term mission that we're now working towards. So one other thing that I wanted to mention is that within Elixir, which is the data research infrastructure for Europe, we're launching a new task force focusing on domestic animal genomes and phenomes. So for the sort of data standards and bioinformatics focused members here, we'd really welcome your expertise and to come and join us in this effort to try and bring together scientists from across Europe within this Elixir infrastructure to coordinate data and technology discussions, to develop and continue to develop metadata standards and develop our metadata standards further, benefiting from the other communities as part of the Elixir consortium, to standardize analysis and workflows, and make very strong connections to the NF Core community here as well, and then take advantage of the organized trainings, workflows, and hackathons that this gives us access to. So if you're interested in becoming part of this effort, it should be launching within the next month. Do get in contact with me and I'll make sure you're in our initial invite list. So really what we've done for the BovReg project, for the EuroFAANG projects and for FAANG globally is gather and present a wealth of standardized data to the community. We've ensured that this is findable, accessible, interoperable and reusable through our use of metadata and ontology standards, customized particularly for BovReg needs, and I thank all of the consortium for all of their effort, particularly early on in the project, in improving those metadata standards so that you can make your data as reusable as possible. And then we've used this to continue to drive the annotations that we produce within Ensemble, and will hopefully enable the first Ensemble cattle regulatory build once we have the data available. And so really what we're going to be doing next is continuing to develop this infrastructure as it becomes part of the EuroFAANG research infrastructure and continue to serve EuroFAANG and global FAANG needs. So I'd like to thank, in particular, the Data Coordination Center team at EMBL EBI. This is led by Alexei Sokolov, who many of you will have had contact with if you've had interactions with submitting data, and then his team, which is Kusim, Milena, and Rahila. And then I'd like to thank all of the EuroFAANG projects for funding, continuing to fund this global effort through the Horizon projects and the future EuroFAANG research infrastructure, which will continue to fund these efforts in the future. Thank you. Any questions? Do you need a mic so that people online can hear? Yeah.
So the question is from Amanda Chamberlain. What data is required to be input to contribute to the regulatory build? Assume it's not FASTQ files. Is it peaks, chromatin states?
Yeah, so it's essentially we need publicly available, so within the European Nucleotide Archive, attack-seq and chip-seq data. So essentially we'll use all of the available data that's in the European Nucleotide Archive. We're essentially waiting because we know the quality of the BovReg data that is coming, and we know the quality of the US data that's also coming as well. We're waiting until we have that available to conduct our regulatory build. But we want to do that before that is ensure that through FAANG we can present the community build that BovReg has done itself so that the community has access to that. And then we hope to then add to that with our own annotation as well, and then present both of them in parallel to the community so they've got the choice of either.
Another question here in the audience?
Krista again. Is there one thing which we previously and currently always discuss is the submission tool? Because I think the BovReg Data Coordination Center is really, is currently mostly fed by the large projects, but it would be nice to have also contributions from many other projects outside, because we know the question had been already in this morning to bring together communities. We can anticipate that there's a wealth of data already also in other communities. Will the research infrastructure, for example, also continue supporting these sometimes demanding submission structures?
Yeah, so my imagination for this is that it will be a combination of both the research infrastructure funding that will continue to fund this, but also EMBL's own funding of this, because EMBL views this as particularly important because Ensemble requires rich data. Without richly described data, particularly for the regulatory builds, one of the things we found that prior to these EuroFAANG projects, the regulatory data was just not well enough described that you could easily and in a timely fashion bring it together in order to conduct a regulatory build. So for us, it's incredibly important that we continue to have rich and well-described data coming into the public archives so that we can produce rich annotations. So it would be a combination of both is what I envisage it to be funded by. As well as we do also get funding from the UK government in collaboration with the University of Edinburgh in order to conduct animal agriculture builds.
Clarify, within the funding that we have now, can we pay for the regulatory build? Is there funding for that? Or do we need to get I guess my main question is, are we relying on the ensemble grant being refunded to be able to do it?
So essentially, the best route of funding would be our UKRI funding. And so that's why it's important, and we thank those reviewers in the community who continually give us very good scores with the UK government. This is, I think, we'll be going for our 4th iteration of this grant. And I think that's a really important grant that supports the work done at University of Edinburgh to allow us to continually to improve the annotations that we have at Ensemble. And that collaboration is very fruitful. So we thank the community to continue to support that, because that's what really allows us to do this. And that would be probably the best effort that we would have to do the cattle build.
Thank you. Yeah, so just to give some context, so that grant runs until 2025, I think September. So, but we need to apply for refunding of that this year. So, yeah.
Yeah. So we'll probably be, Emily and I will be contacting you for support and good references, please.